Hey guys, welcome back. So this is part 2 or episode 5 of Dental Talks. So today's topic would be glycolysis, the main interesting core of the subject. So now glycolysis is basically, as a reminder, the process in which glucose is broken down and we get energy. Okay. Now the sequence of reaction of glycolysis can be divided into 3 large phases. The first is the energy investment phase or the priming stage. Then is the splitting phase, and the third is the energy generation phase. So now these 3 phases, I'll go in depth about them. Now the first, which is the energy investment phase. In this phase, basically glucose is phosphorylated. So it becomes from glucose, it becomes glucose 6-phosphate. So basically phosphorylation is the process by which a phosphate group is added into the glucose molecule. So now at which position is it added? It's added at the 6th carbon. Okay. So this is the glucose structure, and that is where glucose 6-phosphate is added. Now the glucose 6-phosphate is formed using the enzyme hexokinase or glucokinase. Now both of these are isoenzymes. Now this is the first irreversible reaction in the process of glycolysis, and it depends on the presence of both ATP and magnesium. Okay. Now the second part is when glucose 6-phosphate undergoes a conversion into fructose 6-phosphate. Now both are isomers, as in they are very similar to each other in structure. And this happens in the presence of enzyme phosphohexoisomerase. Okay, phosphohexoisomerase, and in the presence of magnesium. Okay, now fructose 6-phosphate is again phosphorylated, as in a phosphate group is added into it, and it becomes 1,6-bisphosphatase by phosphofructokinase enzyme, or PFK in short. This is the second irreversible reaction and is considered as the regulatory step in glycolysis. So which is the regulatory step and which is the important enzyme that regulates glycolysis? It's phosphofructokinase or PFK. Now these 3 were the energy investment phase reactions. Now coming to the second part, which is the splitting phase. Now as we know, glucose is a 6-carbon chain monosaccharide. So the 6-carbon fructose 1,6-bisphosphate, which was the end product of the energy investment phase, is now split, and it becomes 3-carbon compounds called glyceraldehyde 3-phosphate and dihydroxyacetone phosphate. Now the enzyme that is responsible for this is aldolase. Fine. So this is the 4th enzyme. Now the next part is when these 3 carbons undergo a process with the enzyme phosphotriose isomerase. You can remember it as PTI. Okay. With the help of this, it becomes a reversible conversion into glyceraldehyde 3-phosphate and dihydroxyacetone phosphate. So DHAP, DHAP, can you remember it like that? Okay. Now there's 2 molecules of glyceraldehyde 3-phosphate are obtained. One in this step, which is the 5th, and one in the previous step, which was the 4th. Now the 6th part, most important part. Now this is the energy generation phase. And the first step is to convert this glyceraldehyde 3-phosphate into 1,3-biphosphoglycerate. Now how is this done? So in the presence of glyceraldehyde 3-phosphate dehydrogenase. So basically a dehydrogenase enzyme removes a hydrogen group, correct? Now that is how it converts the glyceraldehyde 3-phosphate to 1,3-bisphosphate glycerate. Okay, or you can remember it as 1,3-BPG. Okay, 1,3-bisphosphoglycerate. Fine. Now this step is involved in the formation of NADH plus H plus. So now this is another form of energetic molecules, just like the ATP family, and a high energy compound, that is the 1,3-BPG. Now the 7th step is when the enzyme phosphoglycerate kinase, or PGK is how you can remember it as, acts on 1,3-BPG and results in the synthesis of a molecule of ATP and formation of 3-PG. Now what is 3-PG? 3-PG is 3-phosphoglycerate. Now this step is an example of a substrate level phosphorylation. since the ATP is synthesized from the substrate without involving the electron transport chain. Okay, so now that is the meaning of a substrate level phosphorylation. Now this is the first substrate level phosphorylation. So which is it? Step number 7. What is formed in this? 1,3-BPG is converted into 3PG. Correct? Remember this. Now the phosphoglycerate kinase reaction is reversible in nature and is a rare example among the kinase family, because most of the others in which kinases are involved are irreversible in nature. Okay, just like phosphofructokinase and even hexokinase or glucokinase, which is the first and third steps of glycolysis. Both these kinases were irreversible. But in the 7th step, the kinase which is there, which is phosphoglycerate kinase, is reversible. Okay, you have to remember it like this. Now the 8th step is when 3PG or 3-phosphoglycerate is converted to 2-phosphoglycerate by phosphoglyceratemutase. Okay, now this you have to remember it as 3PG becomes 2PG by phosphoglyceratemutase. So what is happening? A phosphate group is removed. Okay, now in the 9th step, a high energy compound called PEP or phosphoenol pyruvate is generated from the 2-phosphoglycerate. Now the enzyme that is involved here is the enolase enzyme. Now this enolase enzyme requires both magnesium and manganese for its activity and is inhibited by the enzyme fluoride. Fine. Now for blood glucose estimation in the lab, fluoride is added into the blood so as to preserve the cells before blood glucose analysis. Now the last step is where the enzyme pyruvate kinase catalyzes the transfer of high energy phosphate from phosphoenol pyruvate, which is PEP, to adenosine diphosphate. Now this leads to the formation of the final molecule of ATP. This step is again an example of substrate level phosphorylation, and this reaction again, just like all other kinases, is irreversible. This was kind of a long talk, but in the next video I'll explain more about what happens in the next part of glycolysis. Have a nice day. Speak to you soon.